Hello everybody, my name is Maxim Vinikov, and I'm going to be going over what Parvovirus is, and specifically how its mutations in this virus, recent strains, have caused it to be very virulent in the canine community. I'm going to give you guys a general overview of what Parvovirus is, basically what it is, the kinds of infections that we see, treatment plans we have, and disease prevention strategies that we have in the modern day. We're then going to go ahead and dive deep, kind of going over and focusing on its structure, the genetic makeup, and then really looking at the evolution of the different strains and what mutations have caused it to be very virulent in our modern day canine population. So here's some quick data on parvovirus from 2013 to 2018. As you can see, infection rates have remained very steady. The subfamily Parvovirinae is known for infecting vertebrate hosts. Hence, this is kind of important to understand because it only does infect vertebrate hosts. And our first strain was found in 1978. So what exactly is parvovirus? Parvovirus is a virus that infects canines primarily, although infects other species of animals as well. Highest risk group are considered puppies, 4 months old or less, and unvaccinated dogs. Parvovirus specifically targets the gastrointestinal tract, although it can go ahead and infect other tissues. The primary method of infection and spreading is through the oronasal route. This can be done with like any kind of infected feces or surfaces, vomit, diarrhea, as well as even other dogs. Approximately it takes 1,000 virons to infect another dog, but any contact provides high risk. So our infection signs and health kind of effects, again, we have, diarrhea, dehydration, vomiting, huge loss of fluids from the body, and that could create, permanent damage to intestines. It incubates for about 7 to 14 days. It hitches a ride through the lymphatic system and infects lymphocytes where it can hide inside them. And then when it gets to the GI tract, as you can see here, it kind of hides in these little coveys of the GI tract so it can reproduce and not be found. Here are some treatment plans that we have, as you guys can see based on this chart. If there are no treatments for the dog, provided the dog is at very high likelihood that it will die. Currently, we have no drug to kill the virus in a host, but we have lab tests and whatnot and preventative kind of medicine that we can do. If our dog does get infected for, say, whatever reason, we try to support it as best we can with fluids and nutrients. However, we try to support the dog's immune system as much as we can so that it can fight off the infection on its own. So how can it be prevented? Like I mentioned before, vaccinations for puppies between 14 and 16 weeks of age. However, that doesn't mean that you're completely immune. Adult dogs are at risk as well, especially when in contact with those feces. High public areas like kennels and grooming centers is where the virus is known to really spread. Of course, when there's also a small risk of not developing immunity, even when, say, a puppy gets all of its shots and gets vaccinated. Now I'll go ahead and going into our deep dive, specifically the important aspects of what I want to focus on, and that's going to be the mutations that made parvovirus so virulent. Here's a quick overview of the general structure of the parvovirus capsid. It's a T1 icosahedral symmetry. It's got about 20-30 protein capsids in nanometer in diameter. It's got no envelope and it's considered a naked virus, but the biggest thing here, takeaway, is that it can survive 1 year in the environment, and its protein code is very resistant to cold, dryness, humidity, different kinds of bases and acids, and even some solvents. There was a study done that showed that bleach seems to be the only way effectively to kill it off a surface. So the genetic makeup of its structure is that it's a single-strand DNA virus. Based off the Baltimore classification system that we learned in class, it's a group 2 virus. It's non-segmented, it's linear, 5 to 6 kilobase pairs. But the key, again, takeaway here that really increases its virulence are those hairpin telomeres that mediate its different replication stages, when it's active in the host, when it's not active in the host. And these play a huge role in how the virus infects the host and how quickly it can kill the host. As mentioned before, there are different strains of the parvovirus that really kind of contribute to its virulence. The key here to look at is that the CPV2 strain does not exist in the worldwide population anymore. It's kind of died out, and all these other 3 strains have taken over. Specifically, what we're looking at here is the CPV2C strain, which is highly virulent and contagious, and infects, has a high infection rate, especially in the United States. So going down here, you can see that, as I mentioned before, it has originated from other species, and because of these mutations, it has been very good at infecting canine hosts. The sequencing that was done by studies I overviewed showed that it had these close relation to Arctic foxes, minxes, and raccoons. And again, as stated before, it did disappear from the population and was kind of taken over by the 2A, 2B, and 2C strains. So the main mutation here that occurred in this CPV2C strain was on the amino acid position of 426. It went from an ASP to a GLU, and this caused, for whatever reason, for it to be more adaptable to its host, as well have an overall stronger kind of protein coat that allowed it to be more protected in hazardous environments. It is believed that the change from the ASN to ASP has caused a change in the coat, and the ASP to GLU has caused better adaptability to the host. Here are some features kind of that I wanted to go over. Again, these small changes in amino acids have allowed it to target different kinds of tissues as well as the GI tract. And it's also been revealed that while the original CPV vaccine that was created may or may not be quite as effective as we thought before because of these new strains. And these mutations in the amino acid have really allowed it to be better suited for attacking the canine host. So the quick summary that I just wanted to go over is that these mutations in the amino acid strains, as well as the hairpin loops that it has, allow it to really control how it infects the host, when it infects the host, and really allows it to survive in any environments for a long amount of time. And it's currently, the CPV2C strain is currently the biggest threat to our canine populace. Thank you very much, and here are my sources.